Imagine you're a surgeon. You walk into the operating room and your scalpel can tell the difference between a cancer cell and a healthy cell sitting right next to it. Same DNA from the same patient, same tissue, but it just knows. That scalpel, of course, doesn't exist yet, but as of a paper published yesterday in Nature, the molecular mechanism behind it does. And it arrived alongside a finding that might matter even more. A discovery that the reason gene therapies haven't been working as well as they should isn't the therapy itself. It's that nobody thought to feed the cells first. A team from Wageningen University and the Van Andel Institute found something in a heat-loving bacterium that changes what CRISPR can do. The enzyme is called thermocas9, and unlike every other CRISPR tool in the existing arsenal, it has a physical property that makes it sensitive to something called methylation, which is tiny chemical tags that sit on DNA and differ between healthy cells and cancerous ones. So let me make that really concrete. Your DNA and a tumor cell's DNA can be genetically identical, same sequence and same letters. The difference is in the annotations. Methyl groups, little chemical caps attached to specific locations on the strand. Healthy cells tend to have these caps in place, but tumor cells often lose them. It's like two copies of the same book, but one has all of its chapter headers removed. Thermocas9 checks for that chapter header. If it's there, the enzyme physically cannot latch on. The methyl group blocks the binding site, the way that a wrong key can't turn in a lock. But if the chapter headers are gone, as they typically are in cancer in this example, the enzyme grabs hold and cuts. In lab experiments with human cells, thermocas9 cut tumor DNA and left healthy DNA alone. Same patient, same genetic sequence, different methylation and different outcome. What makes this significant isn't just the precision, although that's amazing, but it is the kind of precision. Every cancer therapy we have today that tries to distinguish healthy from sick tissue does it by reading the outside of the cell, surface proteins, receptor shapes. That's how CAR-T works, which has been extremely successful, and we've covered that recently. That's also how antibody therapies work. But thermocas9 reads the inside of the cell, the epigenetic layer, a deeper stratum of biological information that surface scanning approaches literally cannot access. An aberrant methylation isn't just a cancer signature. It shows up in neuroblastoma, autoimmune disorders, and a range of conditions where the disease looks genetically identical to health, but behaves, of course, completely differently. Now, I want to be really clear about where this stands. The study demonstrated selective cutting. It has not yet shown that cutting alone is enough to kill the tumor cells, and that's important to remember. Also, delivery, actually getting the thermocas9 into a tumor inside a living person, remains unsolved. Those are real gaps, and I'm not glossing them over. But the Century Report has tracked the gene therapy arc across several months now, and what keeps emerging is that the hardest problems in this field aren't being solved by building more complex tools. They're being solved instead by understanding biology's existing architecture more precisely. Back on April 10th, the CAR-T therapy that we covered, which was originally built for cancer, was repurposed to eliminate 3 autoimmune diseases in a single patient. Same platform, different reading of what the immune system was actually doing. Thermocas9 follows the same logic. The enzyme isn't engineered to be smart. It's structurally sensitive to a chemical distinction that biology already maintains. And that brings me to a second paper, published the same weekend, that attacks the other half of the gene therapy problem. A team led by Daniel Zhongji Wang and Shana Kelly, published in Science Translational Medicine, essentially caught the entire mRNA delivery field looking in the wrong place. For years, and I mean years, hundreds of labs have been redesigning lipid nanoparticles. These are the tiny fat bubbles that carry genetic material into cells. Every mRNA vaccine you've ever received used those, and a massive amount of effort has gone into making those bubbles better. Better shapes, better surface chemistry, AI-optimized formulation, and clinical results kept underperforming what the lab results would predict. Wang and Kelly's team asked a different question instead. What if the bubble is fine, but the cell isn't ready? They grew cells in a medium that actually resembled human blood plasma, not the nutrient-rich soup that labs typically use. And LNP uptake dropped 50 to 80%. The cells living in conditions closer to what they'd experience inside your body simply weren't metabolically prepared to absorb the delivery vehicles. Think of it like showing up at someone's house with a delivery and the door is locked. You can redesign the package all day, but it won't make a difference. The problem is the door. The solution? 3 common amino acids: methionine, arginine, and serine, administered alongside the lipid nanoparticles. So not a drug, not a novel compound. Amino acids your body already uses every day. This combination increased mRNA delivery up to 20-fold, and it pushed CRISPR gene editing efficiency from roughly 25% to nearly 90% in a single dose. In a mouse model of acute liver failure, survival went from 33% to 100% when the amino acid supplement was added. Same nanoparticle, same genetic cargo, but different cellular environment. Now think about what 25% to 90% means in practical terms. If you're a patient receiving a gene therapy, 25% editing efficiency might mean partial correction of a genetic disease. Some of your cells get fixed, but most don't. 90% means the therapy works. That's the difference between a treatment and a cure. And this wasn't achieved by simply redesigning the therapy itself. It was achieved by recognizing that the biological environment receiving the therapy was the bottleneck. Every lipid nanoparticle formulation currently in clinical trials for genetic diseases, for cancer, for vaccines could potentially benefit from this approach. So this is not a new pipeline. This is an upgrade to the existing one. Now put the two findings side by side. ThermoCast9 gives you a cutting tool that distinguishes cancer from health by reading internal chemical signatures. The amino acid supplement gives you a delivery system that actually works at therapeutic efficiency inside a living body. Neither was built by adding complexity. Both were built by paying closer attention to what biology was already doing. The gene therapy field has been engineering louder megaphones. These teams figured out the room had bad acoustics. Okay, let's shift away from molecular biology to institutional biology, because institutions have their own version of internal contradictions that only become visible when the pressure is high enough. Axios reported yesterday, citing 2 sources with direct knowledge, that the NSA is already using Anthropic's Mythos Preview, the same mythos that the Pentagon designated a supply chain risk. The NSA operates under the Department of Defense, as you know. So the same organizational body that blacklisted Anthropic's products is actively deploying the company's most powerful model through its own intelligence arm. The NSA reportedly received access through Project Glasswing, which is Anthropic's early access program for roughly 40 organizations, and that list is slowly growing. And one source told Axios that the model is being used more widely within the department beyond just the NSA. So the operational logic is transparent. Mythos' cybersecurity capability is too consequential to go without, regardless of what the legal posture says. And the contradiction isn't confined to just one building. Australia's security regulator, ASIC, confirmed yesterday that it's monitoring Mythos, making it the 4th continent where a financial regulator has publicly said that this model requires ongoing scrutiny. The Bank of England, the Fed, the ECB, the IMF, and now ASIC. Meanwhile, Anthropic's Guillaume Francin told AFP the company has tripled its annualized revenue quarter over quarter to more than $30 billion, surpassing OpenAI for the first time. Europe is Anthropic's fastest growing region. Stay on that for just a minute. The company that absorbed the most government pressure, blacklisted, designated a supply chain risk, sued, is now generating the most revenue and receiving the most institutional attention. The Century Report tracked this dynamic starting when Claude hit #1 in the App Store during the Pentagon confrontation. The assumption that holding safety red lines would be commercially punitive has been structurally inverted. The red lines instead became the credibility that opened the doors that now generate the revenue. And the governance picture this creates is genuinely new. Financial regulators across 4 different continents coordinating their monitoring. An intelligence agency deploying what its own department banned. The OMB preparing access for Treasury, Commerce, Homeland Security, Justice, and State. What we're watching isn't institutions catching up to a new technology. It's institutions improvising governance in real time through contradiction, because the capability arrived before the framework did. And that's not a failure of governance. It might be what governance becomes when the ground never stops shifting. Messy, contradictory, and faster than design. Okay, let's shift away again. MIT Technology Review published a detailed account yesterday of something that has no clean historical parallel. Chinese tech workers are being told by their employers to document their own jobs in enough detail for an AI agent to replicate them. And a counter-movement is building tools to sabotage that process from the inside. Since OpenClaw proliferated across Chinese workplaces, employees report being pushed to catalog their workflows, every decision tree, every judgment call, every routine, into formats that an AI agent can consume. The viral GitHub project Colleague Skill, created by an engineer at Shanghai Artificial Intelligence Laboratory, formalizes this. It claims to extract a coworker's duties, personality, and work patterns from chat histories and files, producing a reusable AI agent manual. And then Koki Su, a 26-year-old AI product manager in Beijing, published the counter tool, an anti-distillation skill on GitHub that rewrites workflow documentation into generic, non-actionable language. Users choose light, medium, or heavy sabotage depending on how closely their manager is watching. The video explaining it got more than 5 million likes across platforms. So what's remarkable about this is who's doing the sabotaging. These aren't factory workers unfamiliar with this technology. Xu is an AI product manager. The Colleague Skill creator works at an AI research lab. The sabotage tools are sophisticated prompt engineering countermeasures that require deep understanding of how language models parse documentation. The people being replaced are fluent in the exact technology replacing them. Here's something I think is genuinely important. Every prior technological displacement, weavers, switchboard operators, typesetters, happened to workers who understood their craft but couldn't articulate precisely where the machine stopped and human judgment began. This displacement is different. The act of resisting, of choosing which tasks to document faithfully and which to deliberately obscure, is producing the most granular record ever created of what human judgment actually consists of at the task level. Which workflows collapse into mush when stripped of context? Which resist codification even under heavy sabotage mode? Which require the worker to be present in ways that no document can transmit? Previous transitions destroyed tacit knowledge silently. This one is being forced to name it, skill by skill, in machine-readable format by the people whose livelihoods depend on proving it exists. And while that labor friction plays out in Chinese offices, a different kind of friction is reshaping American elections. NPR published a national investigation yesterday confirming that data center opposition has crossed from zoning disputes into midterm electoral politics. Voters in Festus, Missouri unseated every incumbent council member over data center approvals. In Independence, Missouri, 2 more council members lost their seats for the same reason. Near Meta's Louisiana data center, residents report brown water and disinfectant smells since construction began. Cities are moving public meetings outdoors because the crowds don't fit inside anymore. The Century Report has tracked this arc since the Port Washington referendum, where voters required direct approval before data center tax breaks could be granted. Since the Indianapolis council member's home was shot at, with a no data centers note left behind. What NPR confirmed is that these are no longer isolated incidents. This is a political category. One policy expert quoted in the piece said it very plainly. It has become a kitchen table issue. And there's an energy story sitting right beside it. The IEA's 2026 Global Energy Review, released over the weekend, confirmed that solar was the single largest contributor to global energy supply growth in 2025, more than 25% of the total increase. Solar is no longer the promising alternative. It's the leading source of new energy on the planet. The irony is sharp. The intelligence era runs on electricity. The fastest growing source of that electricity is solar, and the physical infrastructure that connects the two, the data centers, is getting rejected by community after community because the costs land locally, while the benefits distribute globally. What emerges from that friction will determine how the abundance the energy transition is building actually reaches the people living next to the hardware. Okay, just a few notable releases from yesterday. Qwen shipped Qwen 3.6 Max Preview, which is a new model live now in their chat interface, continuing the cadence of Chinese frontier releases. And a few days ago, Minimax released Max Hermes, which is a hosted, always-on Hermes agent that can learn from its own work, remember across sessions, and run long-horizon automations without you standing up any infra. So this is a long-term cross-session agent, not just a chat. If you're someone who likes the idea of Hermes OpenClaw but doesn't really want to deal with Docker, infra, model routing, and cron wiring, this gives you a managed click-to-use version, and it's available now. Okay, here's what to carry from today. 2 papers published the same weekend solving different halves of the same problem. One teaching a molecular tool to tell cancer from health. The other teaching a cell to accept the therapy that's been knocking on its door for years. An intelligence agency deploying the model its own department banned, while regulators on 4 continents figure out oversight in real time. Workers documenting exactly which parts of their judgment can survive extraction and which can't. Voters throwing out officials over the physical footprint of a digital era. Now, very soon, watch for whether that amino acid supplement moves into human trials, because if the mouse results translate, then every mRNA therapy in the pipeline just got a potential multiplier. Watch for the next Mythos governance development, because the gap between the Pentagon's legal posture and the NSA's operational reality can't hold forever. And pay attention to what happens in those Chinese GitHub repositories, because somewhere in the difference between what workers faithfully document and what they deliberately obscure is the sharpest map we've ever had of what makes human work human. See you tomorrow.